Thank you very much. I want to thank Yaniv and all the organizers and NSF and BSF for bringing us all here, finally in person. It feels especially great to be in this very special place in Israel after years of pandemic Zooms. So my lab builds computational models fed with parameters all extracted from experimental data in order to test neuroscience theories. And I will illustrate this approach with a very famous region of the brain, the mammalian hippocampus. So the mammalian hippocampus has long been involved in the consolidation and retrieval of episodic memories. For example, you go to a conference after a few years, you see a colleague and you retrieve in your mind the experience of having met that colleague at a conference maybe 5 years prior. And this process of episodic memory in cognitive science is known to consist of 3 distinct processes: pattern separation, pattern completion, and pattern comparison. And the anatomy of the circuit of the hippocampus has been known for over 100 years, and it's conveniently divided in 3 synaptic stations, the dentate gyres, CA3 and CA1. And according to textbooks, each of those is responsible for the performance of one of those. So the dentate gyrus for pattern separation, where you decompose the experience in individual elements, such as the identity of a given colleague from a crowded scene. Pattern completion, where you use that clue and that cue to retrieve a complex memory from your past. And pattern comparison in CA1, where you analyze both the differences and similarities between those experiences. And particularly, pattern completion in CA3 has been hypothesized to occur thanks to the very dense recurrent collaterals between the axons and the dendrites in this region, and has been formalized by seminal mathematical models, especially the Hopfield net. But the question remains, does this function, or how can this function occur in a biologically realistic circuit or in a real circuit of biophysically complex neurons and synapses? So in order to answer that question, we built a model in silico of CA3 in the mouse in real scale with approximately 80,000 neurons. Each of the neurons corresponded to a type of neuron, one of 8 types, which were morphologically and chemically identified. The principal cell of this region is a pyramidal cell, which is excitatory, and there are 7 known types of inhibitory GABAergic cells, each with their own specific connectivity and function in the circuit. And for each of these neuron type, we have the input-output function, which is modeled with a dynamical model system called Izikevich neurons, quantitative probabilities of connection, as well as experimentally fitted synaptic physiology for the communication between neurons using the Thodek's Markram formalism. And finally, we have long-term plasticity in the form of Hebbian symmetric STDP between excitatory synapses. So all the parameters were taken from a very powerful open access resource called hippocampum.org, which is based on over 50,000 experiments where all the parameters are accessible online, and the simulations were run on graphical process units using the environment called CarlSim. And when I say we, I really mean my wonderful PhD student, Jeffrey Kopsik, who will graduate next year and will be on the postdoc market. He comes greatly recommended. So to summarize, the model has 8 neuron types, 51 connection between neuron types, and when you implement it in a network, it boils down to 80,000 neurons, 250 million synapses, and the number of parameters experimentally determined is 386. To illustrate the process of what those parameters are, let me take the example of just 2 neuron types in this, a CA3 pyramidal cell and a CA3 basket cell, an inhibitory cell type. So there are about 75,000 of the first and 115 of the second in a mouse. And for each of those, you have the probability of connection, in this case, both between pyramidal and pyramidal, between pyramidal and basket, and vice versa, as well as the excitability of the neurons, so the response of the neurons to a given synaptic stimulus that is fed to the experimental data. And lastly, the synaptic physiology of how a neuron responds to a series of spikes. And in this example, the excitatory postsynaptic currents in the basket cell and the inhibitory postsynaptic current in the pyramidal cell. So when you put together this network in real scale and you let it go, it actually behaves in a very interesting way. There are chaotic dynamics which are stable. The network never turns off, never saturate. In this case, it's an example of about 700 or so neurons with a roster. Each dot in the upper panel is a spike. And the firing patterns that the neurons go into are reflective of their in vivo patterns. So pyramidal cells have very sparse firing. Basket cells and axonic cells are fast spiking. And the individual voltage traces of those example neurons in this case also are reflective of in vivo activity. And this was work that we published last year in cognitive computation. So we're in well positioned with this toy model to then say, can we now study pattern completion? So this is the seminal theory of assembly formation. So the way that the protocol works is that you start with all these, in this case, 75,000 pyramidal cells and their connectivity, and you take a random set of, in this case, 300. We started with that number. We declare that as an assembly, and during training, we activate together those 300 cells in systematic ways. And after a few training sessions, we test it by, as an example, only activating a subset of the cell assembly and see if the rest of the assembly reconstructs. So in order to use, again, biological inspiration, we do the training locked into theta rhythms, which is one of the natural rhythms of the hippocampus. So each assembly is trained at every theta. So this is about 4 times a second. And then during testing also is tested in theta. Within a theta, if you look at the bottom right panels, the coactivation of subsets of neurons in the cell assemblies occurs during gamma cycles, so at higher frequencies, again, inspired by the biological data. And if you look at the top right panel, which is the theta testing regime, in this case, we only activated half of the assembly of each of 3 assembly, and in all cases, the non-activated neurons respond to that cue. So how can we quantify that systematically? The first metric is really basically doing what I just described, which is you only activate part of the assembly and you measure pattern completion by looking at whether the non-activated neurons within the assembly behave more like the activated ones or behave like the non-assembly neurons. And you can express that mathematically using a z-score. Another alternative is to use a different protocol, which is also sometimes called pattern completion. To avoid confusion, we call that pattern reconstruction. And this is the protocol that is used, for example, in the experimental lab of Peter Jonas in Austria, where his postdoc, Guzman, basically distorts the activity of neurons in the mouse. So in this case, instead of only activating part of the assembly, a subset of those 300 neurons, we still activated 300 neurons, but not all as part of the assembly. Only a subset is part of the assembly, and the rest is noise from non-assembly neurons. And then we look at whether the response of the network is more similar between testing and training than the stimulation of the network. So those are 2 metrics to measure pattern completion. And let me show you some example results. In this case, it's a run of the full spiky neuron network. If you look at the very first portion, the very first about 700 milliseconds, the network turns on. It takes about 700 milliseconds for the network to achieve a steady state. The first period of about 3/4 of a second after that is a resting state where we let the network go into spontaneous rhythms. And then we start a very brief training section, in this case of only about 1.5 seconds. So this is a very small setting of only 10 training sets. And after that, we let the network rest again, let it do whatever it does spontaneously, and then we go into testing mode where we only stimulate part of the assemblies. And just to enlarge only the assembly component of that testing period, we show again, in this case, the red are the stimulated neurons. It's only 60 out of 300 in each assembly, so only 20% of the assembly after a very brief training protocol. And the remaining 80% of the cells respond after about half of the theta cycle to complete that pattern in a very effective way. And this is done, in this case, for 3 assembly. Now, if you just look at the number of CA3 pyramidal cells In the mouse, there is space for about 250 independent assemblies to be trained and retrieved in this way, but that's entirely with no overlap. In reality, it is almost certain that assemblies do overlap in the real hippocampus, and we're now well positioned to study the effect of overlap on memory capacity and performance. So the overall summary of all the results is that we can obtain both pattern completion and pattern reconstruction in very robust way over a wide range of pattern degradation. So we go from only 10% of the assembly that is stimulated as a clue to 90%, and the number of presentations go to as little as 10 to up to 100. And in most cases, we get pattern completion well above 50%, closer typically of 70 to 98% or so. So this is the result from the first year of our grant. The next step will be to in fact investigate the role of the individual GABAergic neurons. Specifically, there are very vague theories about what the perisomatic cells, basket cells, and axonic cells do, and the dendritic targeting neurons, typically OLM cells, IV cells, and mossy fiber associated cells, and what they do on both the training and the testing regime. Last but not least, my acknowledgement, this grant was generously funded by the Department of Energy with Program Officer Dr. Robinson Pino, and my partner in crime is Professor Gina Adam, who is actually working on the neuromorphic implementation of these kinds of spiking neural network in hardware, and she has a poster that, despite what the program said, is now hanging in post number 43 upstairs. So I hope to see you there during the reception tonight, and I'll stop here.
Questions?
So I have three small questions. One, what is the factor that you gain in your simulation when you use GPU versus CPU? This is the first. Second, why you are using Izhikevich neurons, and how you do introduce their electrophysiology, and whether the electrophysiology means also reconstruction of the special structure of the neurons. And third, what is the role of these electrophysiological properties and the diversity between the different populations in the pattern completion?
Awesome, wonderful questions. So the first one about GPU, it's just a matter of speed. So we can run a network with about 80,000 neurons. I wish I could say real time. It's not quite real time, but only about 1 in 10. So that means that we can run many simulations per hour. In fact, much of the time is just to implement the simulations, then they run quite quickly. So you could not possibly do that with CPUs unless you can afford really large supercomputer clusters. The second one is why Izhikevich model. So Izhikevich models are computationally very efficient, and they are known to be able to reproduce the very wide diversity of firing. Specifically in the hippocampus, you have dynamics that go from adaptive spiking to regular spiking to bursting to stuttering, etc. And by simple tuning of those parameters in the Izhikevich models, you can reproduce them all. And what is the role of that diversity in the circuit? Each of the neuron types that is characterized has its own unique signatures of firing patterns. And although the Izhikevich models are simplified models, these can be casted in as what we call simple compartmental models. So each neuron might have 4 or 5 compartments. Each compartment is one of the layers of the hippocampus. So the connectivity that each neuron type receive or send to other neurons is compartment specific. And this, in fact, allows to fully reproduce what we know about the in vivo firing patterns in the neurons.
Other questions?
Generate local field potential from this model?
Yes, in fact we can. So the question was whether we're able to generate local field potential. The answer is yes. In fact, we're very interested because just at the last Society for Neuroscience about 9 months ago, one experimental lab has been able to measure local field potential in CA3. CA3 in the rodent is a deep structure, so it's much harder to record than CA1, but now we are, I think, well positioned to try to see if that emergent property is reproduced.
Okay, thank you. Thank you for